Fluorescence in situ hybridization, FISH, is a molecular cytogenetic technique used to detect and localize specific DNA or RNA sequences within cells or tissues. It combines nucleic acid hybridization with fluorescent microscopy, enabling visualization of these sequences within their cellular context. FISH can be broadly classified into two main forms, DNA-FISH and RNA-FISH. DNA-FISH targets specific DNA sequences to detect chromosomal abnormalities, gene copy number changes, and structural rearrangements, whereas RNA-FISH targets RNA transcripts to investigate patterns of gene expression. Regardless of whether DNA or RNA is being analyzed, FISH follows similar general steps. Cells or tissue sections are first fixed onto microscope slides to preserve cellular morphology, prevent degradation, and immobilize the cells on the slide. The cells are then permeabilized to allow fluorescent probes to enter. For DNA-FISH, the DNA is denatured to separate the double-stranded DNA into single strands, whereas RNA is already single-stranded and generally does not require denaturation. Fluorescently labeled probes specific to the target sequence are then applied and hybridized through complementary base pairing. Probes may be directly labeled with fluorophores such as fluorescent, FITC, rhodamine or cyanine dyes, CY3 and CY5. Alternatively, they may be indirectly labeled with molecules such as biotin or deoxygenin, which are subsequently detected using fluorescent reporter systems. Multiple probes labeled with different fluorophores can also be used simultaneously, allowing several target sequences to be detected in a single experiment. Following hybridization, excess probe is removed by washing and the samples are examined using fluorescence microscopy to detect the presence and localization of the target sequence, and in DNA-FISH applications to assess gene copy number. FISH can be performed on either metaphase chromosomes or interphase nuclei depending on the application. Metaphase FISH provides high-resolution visualization of chromosome structure and the chromosome location of target DNA sequences. One important application is chromosome painting, in which differently colored probe sets are used to visualize entire chromosomes or large chromosomal regions simultaneously. Interphase FISH is faster and widely used clinically because it does not require cell division or cell culture, making it the preferred approach in many diagnostic laboratories. In the clinical setting, DNA-FISH is widely used in prenatal testing and in the diagnosis of both solid tumors and hematological cancers. In cancer, it is performed on tumor tissue in solid cancers or on blood or bone marrow samples in hematological malignancies to detect somatic, that is, acquired genetic alterations present in cancer cells rather than the inherited germline variants. Accurate detection therefore depends on appropriate sample selection to ensure that representative cancer cells rather than the surrounding normal tissue are analyzed. While RNA-FISH has increasing applications in clinical settings, it has primarily been used in research to investigate gene expression, RNA localization, and cellular heterogeneity. Overall, FISH remains an indispensable tool in modern cytogenetics because of its high sensitivity, specificity, and ability to analyze genetic material within intact cells. Its versatility in detecting chromosomal abnormalities, gene copy number changes, structural rearrangements, and RNA transcripts continues to make it invaluable in both biological research and clinical diagnosis. Do try the accompanying questions to test your recall and understanding of the topic. If you found this video helpful, please like and subscribe to support the channel. Thank you for watching.